If you have type O positive blood, you're carrying the single most common blood type on Earth. More people share your blood type than any other, roughly 35 to 38% of the entire global population. And in some regions of the world, that number climbs even higher. For decades, that fact got treated as a coincidence, a simple statistical majority with no real story behind it, the biological equivalent of being the most common eye color or the most common shoe size, unremarkable simply because it's everywhere. Then researchers went looking for why O blood became so dominant in the first place, tracing the question back through disease records, ancient bones, and blood samples collected from opposite ends of the Earth. What they found wasn't one story, it was two, playing out on opposite sides of the planet for almost completely unrelated reasons, both converging on the exact same blood type sitting in your veins right now. This is where type O blood actually comes from, why it became the world's most common blood type, and why the real explanation splits into two entirely different survival stories depending on where your ancestors lived. For most people, blood type gets filed away as simple biology, something learned once in a hospital waiting room or a high school health class and rarely thought about again. You inherit it, you learn it for medical reasons, in case of surgery or transfusion, and beyond compatibility charts, it doesn't seem to mean much else. O positive specifically gets treated as unremarkable, mostly because it's so common, too widespread to seem like it needs an explanation, the kind of trait so ordinary that nobody thinks to ask where it actually came from. That assumption misses one of the more compelling discoveries in recent evolutionary medicine. Here's the first real crack in the idea that blood type is just random biological noise. In 2007, a research team led by Alexandra Rowe at the University of Edinburgh, working with colleagues studying malaria patients in Mali and Kenya, published a study in the Proceedings of the National Academy of Sciences that had been building toward one specific question. Malaria has killed more humans throughout history than almost any other disease, shaping human genetics in the process. Certain blood-related traits, like the sickle cell mutation, are already well known to offer partial protection against it, a genuine evolutionary trade-off scientists had documented decades earlier. Did ordinary ABO blood type, the same system used every day in hospitals worldwide, play a role too? The team studied red blood cells infected with Plasmodium falciparum, the parasite responsible for the deadliest form of malaria, and focused on a specific, dangerous behavior the parasite triggers called rosetting, where infected cells clump together with healthy, uninfected cells, forming small clusters that can physically block tiny blood vessels and trigger the severe, often fatal complications of cerebral malaria, including seizures, coma, and organ failure. When the researchers compared rosetting behavior across all 4 ABO blood types, using blood samples collected directly from malaria patients being treated in the study hospitals, the pattern was unmistakable. Type O red blood cells form dramatically smaller, weaker rosettes than types A, B, or AB. The parasite simply couldn't build the same dangerous clumps inside a person with type O blood, and patients diagnosed with severe malaria in the study were significantly less likely to have type O blood than patients with milder, uncomplicated cases, a statistically clear pattern that held up across the entire dataset. Here's the deeper layer explaining why this matters so much. Malaria has been one of the single strongest selective forces acting on the human genome throughout our entire history, killing enough people, generation after generation, in tropical and subtropical regions to reshape entire populations' genetics around surviving it, in the same way it shaped the spread of sickle cell trait, thalassemia, and several other well-documented blood adaptations across malaria-affected regions of the world. In a disease environment lethal enough, even a partial survival advantage compounds dramatically over hundreds of generations. The same mathematical principle that lets a tiny percentage point edge in survival odds reshape an entire population's genetics given enough time. If type O blood meant even a modestly better chance of surviving a severe malarial infection long enough to have children, that single advantage, repeated across thousands of years in malaria-heavy regions of Africa, the Mediterranean, and parts of Asia, would be more than enough to explain why the O allele spread as widely as it did across large parts of the world, without requiring any single dramatic event, just steady, compounding pressure applied over an enormous span of time. Picture a small village somewhere in West Africa, several thousand years ago, during the height of malaria season. Mosquitoes thick in the humid evening air, children falling sick with fevers that, for many, would prove fatal within days, families bearing more of their young than any modern parent could imagine losing. In that village, families carrying type O blood are quietly surviving severe infections at a measurably higher rate than their neighbors, not through any conscious choice or visible trait, no way for anyone to see it coming or explain it at the time, but through a difference in how their red blood cells behaved once the parasite got inside them. Nobody in that village had any way of knowing why some families kept recovering while others didn't, why certain bloodlines seemed to weather the worst malaria seasons a little better than others, generation after generation. Over enough generations, in enough malaria-stricken villages across enough of the ancient world, stretching from West Africa through the Mediterranean basin and into parts of South Asia, that quiet, invisible advantage was strong enough to reshape which blood type became the most common on the entire planet. Here's the twist that turns this from a single clean explanation into something far more interesting. Malaria explains why O blood spread widely across large parts of Africa, Asia, and the Mediterranean. It cannot explain one of the most extreme concentrations of type O blood found anywhere on Earth, because that population's ancestors never lived anywhere malaria could reach them. Indigenous populations of the Americas carry some of the highest frequencies of type O blood ever measured anywhere in the world. Studies of the Quechua people in the Peruvian highlands have found frequencies approaching 100%. The Maya show similarly extreme concentrations, with some studied groups registering 98% or higher. The Navajo in the American Southwest show markedly elevated O frequencies as well above the global average. This wasn't caused by malaria because Plasmodium falciparum didn't exist anywhere in the Americas until European contact. Thousands of years after these populations had already become overwhelmingly type O, ruling out the exact mechanism that explains O blood spread across the Old World. The real explanation here is a founder effect, an entirely different genetic mechanism than the one driving O bloods spread across the malaria belt of the Old World. When early populations crossed from Siberia into the Americas via the Bering Land Bridge, likely in small, genetically limited groups numbering in the hundreds or low thousands, rather than anything resembling a large migrating nation, whatever blood type alleles that small founding population happened to carry became the genetic starting point for every indigenous population descended from them. If that original founding group carried mostly the O allele, whether by chance or because A and B alleles were simply rare or absent among them to begin with, every population descended from that founding group would inherit the same overwhelming O bias, amplified further by generations of relative isolation across two entire continents, with no later waves of migration bringing A or B alleles back in to dilute it, unlike almost every other populated region on Earth. Ancient DNA extracted directly from pre-contact skeletal remains in eastern North America confirms this pattern was already firmly in place before European contact ever began, ruling out later colonial-era disease epidemics as the cause and pointing instead to that original founding population thousands of years earlier. Two completely different evolutionary stories, playing out on opposite sides of the world, for entirely different reasons, arrived at strikingly similar results. In one, active disease pressure sustained over thousands of years pushed a population toward O blood because it offered a genuine survival edge against one of history's deadliest diseases. In the other, a small founding population simply carried the O allele by chance, and isolation preserved that starting point almost untouched for millennia afterward. Selection in one hemisphere, pure genetic chance, and isolation in the other. Same blood type, arrived at through almost opposite mechanisms. It's worth adding one detail that keeps this story honest rather than tidy. Not every indigenous population fits the pattern cleanly. The Blackfeet and blood tribes of the northern Great Plains show unusually high frequencies of type A blood instead. A genuine exception researchers still don't fully understand. A reminder that founder effects are, by their nature, somewhat random, dependent on exactly which individuals happen to found which population, carrying whichever alleles they happen to have, rather than following some grand, predictable rule. Science rarely hands over a story this clean without at least one loose thread still hanging. And that loose thread is worth remembering before treating any single explanation as the whole picture. So, if you have type O positive blood, here's what your DNA is actually telling you, and the honest answer depends entirely on where your ancestors were living thousands of years ago. If your roots trace back to malaria-heavy regions of Africa, the Mediterranean, or South Asia, your blood type may carry the quiet genetic memory of countless ancestors who survived a disease that killed enormous numbers of the people around them. An active, ongoing battle written directly into your red blood cells. If your ancestry traces back to indigenous peoples of the Americas, your blood type instead reflects something closer to a genetic snapshot, a small founding population's starting deck of cards, preserved with unusual clarity across thousands of years of relative isolation on two continents cut off from the rest of the world's genetic traffic. Both stories are real. Both are documented in peer-reviewed research, built on evidence ranging from modern hospital blood samples to DNA extracted from bones over 1,000 years old. And both happen to end in the exact same three letters on a blood donor card, a coincidence of biology that turns out, on closer inspection, not to be a coincidence at all, but two entirely different chapters of human survival that simply happen to arrive at the same destination.